In the name of Allah, the most beneficent and the most merciful, I am Khadija Rahman from Plant Ecology and Conservation Lab, and under the supervision of Dr. Shafal Herbal on the comparative approach to evaluate species diversity and abundance in the existing ecological zones of Islamabad, Pakistan. As we know that vegetation is an expression of plant species that is mainly affected by different drivers such as topography, morphology, climatic conditions, anthropogenic activities, and other biotic and abiotic components. The disparity in the pattern of species distribution within naturally occurring dynamic habitats can result from semi-humid zone along streams, which offers a unique and useful model system. So the environment plays a crucial role in the sustainability of various life forms and the ecological patterns across an ecosystem. As we know that terrestrial vegetation plays a fundamental role in relating land, atmospheric system, and interaction between them. So this correlation in the urban ecosystem reveals diverse essential processes that flourish variability in vegetation patterns. The communities along water bodies establish a strong bond between biodiversity and ecosystem functioning. The objectives of this study was to evaluate vegetation of two distinct zones, that is the semi-humid zone and the dried zone, to determine the species richness and examine species diversity, to analyze soil samples collected from research study to know about the impact of environmental factors on vegetation of both the zones. This is the study design in which the quadrate quantitative ecological technique was used, that is the randomized quadrates were laid down, 126 quadrates were laid in 21 stations. The distance between two stations, that is station 1 and station 2, were kept 1 kilometer, but the distance between two quadrants were kept 200 meters. The A quadrant denotes the semi-humid quadrant, while the B denotes the dried quadrant laid in the dried zone. These are the materials used during the studies, that is rope, cutter, scissor, tags, newspaper, polythene bags, plastic bottle for keeping the samples, the newspaper for keeping the plants dry, wiring tape, soil digger for soil collection, camera for capturing the pictures of plants, plant presser, umbrella, and pen and marker. These are the chemicals used in lab during the research work, that is mercury chloride, distilled water, nitric acid, sulfuric acid, ethanol, and perchloric acid, perchloric acid. Methodology, the vegetation sampling were done in the field. So for that purpose, quadrates were laid in which plants were collected and the plant species were photographed and were identified with the help of agronomists and ecologists of Kailasan University. Then these were dried and poisoned via mercury chloride and then were mounted on a standardized herbarium sheet. The soil sampling were also done. First of all, the soil samples were collected. Then the soil were mixed and the gravels were removed. Then the soil samples were kept in the polythene bags and were labeled properly according to the quadrate size to know about the sample, and then were analyzed via various meters, that is the pH meter. pH were analyzed, EC was analyzed, and TDS meter were used to know about the total dissolved solids. Then floristic checklist of both zones were evaluated. Then the species abundance and species richness were calculated. And the data was sorted in an Excel. That is the digestion process of soil, in which the soil was in the soil digestion were done in the plant ecology and conservation lab, and then the digested samples were sent to the biochemistry lab for atomic absorption spectrometry to know about the various environmental variables, that is nutrients, macronutrients, and micronutrients. The results revealed that 201 plant species were recorded from the whole area, which belongs to 71 families. As we know, as the area distinguished in the semi-humid zone and the dry zone, so 137 plant species were belonging to 52 families in the semi-humid zone, while the species diversity were higher in the dry zone, that is 152 plant species were recorded belonging to 59 families. The species richness were higher in the semi-humid zone, that is 1965 species were recorded, while the IVI were also higher in the semi-humid zone, that is 35983.3 IVI were recorded in the semi-humid zone, followed by the dry zone. The tree diversity revealed that 18% were trees, 17% were shrubs, while 65% were the herbaceous diversity. Herbaceous diversity were higher in the dry zone, while the abundance of herbs were observed in the semi-humid zone. That is 1475 individuals were recorded in the semi-humid zone. Species abundance and in the dry and semi-humid zone. As you can see here, in the species richness were higher in the semi-humid zone, were followed by 1767 species, that is in the dry zone, while the species diversity were higher in the dry zone, belonging to 59 families. 152 plant species were recorded belonging to 59 families, that was also higher in the dry zone. As you can see, the same number of trees were recorded in both the zones. Almost the tree abundance were higher, only one individual were higher in the semi-humid zone, while having 130 species in the dry zone. The shrub diversity were almost same, but as you can see here, that in the shrubs were higher, the shrub diversity were higher in the dry zone. The herb diversity were higher in the dry zone, but the abundance were higher in the semi-humid zone. That is 1475 individual herbs were recorded in the semi-humid zone, while 123 and 1243 individuals were recorded in the dry zone. The family chart reveals that Poaceae, Asteraceae, and Rhodiosae were the most dominant families, followed by Moraceae and Lamiaceae in the semi-humid zone, while Poaceae, Fabaceae, Convolvulaceae, and Forbiaceae were the dominant families of the dry zone. The dominant trees of zone A are as follows, that is Sapiens sabiferum, that is in the semi-humid zone, Sapiens sabiferum, Melatus philippensis, Rochionecia prepirifera, and Albergia sisu were the dominant tree species of zone A, that is semi-humid zone. Cassia fistula, Borosonacea, Phoenix dactylifera were the dominant species of the dry zone. This graph revealed that Arunditonex, Medium Oleander, and Lantana camara were the dominant shrub species of semi-humid zone, while Bacopa moneri, Paspalum, and Persicaria glabra were the dominant herb species of the semi-humid zone. The dominant shrub species of the dry zone are Lantana camara, Editoda veseca, Cerisa opaca, while the dominant herbs of the dry zone were the Smithseckia bipinnata, Sonodon dactylon, Cymbopogon distans, and Malvestrum coromandelium. These are the species having respective IVI. The Persicaria glabra of the semi-humid zone showed 1000 IVI value, while dominating the herbaceous layer of the semi-humid zone, followed by Pascaline, Cynodon, Dactylon, and the Cotomoneri, while in the shrub species Arundodonyx with 2292 IVI, followed by Nerium oleander, Lantana camara, and Ipomia, while the Berger sisu, Dosonecia pepperifera, Melotus philippensis, and sabiferum were the dominant tree species. The dominant plants of zone B reveal that Marvestrum coromandelium having 800 IVI, followed by Cynodon dactylon 783 IVI value, followed by Cymbopogon distans and Dismostachia, while Lantana camara, Edapota, and Carissa were the dominant shrub species of the dry zone, while the dominant tree species having IVI 1351, that is Phoenix dactylifera, having the IVI 1351, followed by Grossonecia pepperifera 1004, and Cassia fistula having 788 IVI value. So the study revealed that the ecological information on phytodiversity and impact of species richness, it revealed that this environmental variable is the prime factor that controls the diversity and abundance of species, which should be explored more for detailed taxonomic, microecological, and evolutionary study in the region. That is, in the semi-humid zone, the pH, moisture, EC, and TDS were higher, as well as the species richness were higher, that the herbaceous layer were dominating in the semi-humid zone, while in the dry zone, the species diversity were high, as you can see over here. I would like to acknowledge Qaid Azam University Islamabad for providing me a chance to do, and for arranging this conference to present my work over here. through this platform. And I would also like to thank the Plant Ecology and Conservation Lab and Dr. Shujal Khan for facilitating me and supporting me throughout my research work. Thank you so much.